Okay, so it looks like we're rolling. This is going to be your notes that you will be able to use for your Unit 10.2 activity that you'll see on Google Classroom. And I'll also post the notes separately. I just kind of wanted to point out some of the important information as we go through. All right. So your first slide is just about your introduction, just the uniqueness of fingerprints and the primary units that make those up, things like that. Then it gets into the structure of the DNA and what the nucleotide is made out of. The importance here is your nitrogenous bases, which are adenine, guanine, cytosine, and thymine. You're going to see that in an activity that will be posted for you on Wednesday. For the structure, it gives you the whole double helix thing, which you should be familiar with from biology. And then it talks to you right here with the nitrogenous bases about what pairs with what. Again, that's going to be very important on your Wednesday activity to remember that adenine and thymine go together and guanine and cytosine go together. So you'll have just the letters A, T, C, G, and you need to remember what pairs with what. For your typing, it's converting it into a series of bands so you can actually create a profile that you can match to a suspect. Then it's going to go through 3 types of typing: PCR, STR, and mitochondrial. And then you'll see in your question, your Google question that gets posted, that one of your prompts addresses those 3 types, and I think it asks you to compare and contrast. So you'll just want to kind of go back through there, read up on each of those 3 types. What do they have in common, if anything? What is that? What's different about them? So just kind of formulate that into a good response. And then it talks about electrophoresis. Again, you're going to actually have an activity over that on Wednesday. So you probably want to read over that slide before you get to Wednesday, but nothing for your first day of assignments to do with that. Then it just talks about CODIS, which is your database. Remember, all the evidence really that we've looked at up to this point has some form of database that goes with that. So this is the one for DNA. And then it talks to you about all the sources of DNA. So you've got multiple sources here. I do want you to also, part of your reflection activity for this will be to pick 2 of those sources. And I want you to think about it and talk about in your response, where would you find those? What type of crime would you find that in? And you're going to elaborate on that in your response. And then it talks about collecting and packaging. Again, kind of the same stuff, always photograph first, and talks about maintaining the integrity of that evidence. So these are your notes for 10.2. Again, I will post these along with this video so that you can use them on the reflection activity for these notes. All right, thank you.